Okay, so we're going to open this up to the audience, but I just want to get us started with an initial question from me, because I'm genuinely curious what all these folks think. So my lab has been working a lot on sort of clinical applications of single-cell RNA sequencing, for example. We're really interested in doing expression profiling in tumor biopsies and solid tumors and things like that. And I was just wondering if I get some perspective from the panel on, what you think the challenges are, what the obstacles are, and how you might be trying to overcome those in your own work. The application of single-cell omics technology in either clinical samples or in preclinical research.
Do you have any opinions, Peter, before I start?
No.
Well, this is going to be wonderful. I think I would say that there's a number of different problems that come to bear when you start to think about sequencing of clinical samples. Obviously, the first thing to think about is how you're actually going to process the samples. A lot of things come to you in very difficult formats. Sometimes they're difficult to dissociate. Figuring out the appropriate protocols so that you can go through and isolate out cells is important. Maintaining an understanding of whether or not what you've dissociated actually matches what you originally had in the tissue is important, and seeing what you get if you carry through your protocol also makes a lot of sense, in addition to minimizing the time along those steps. So in a way of not just saying that these are problems instead of more giving solutions, we spend a lot of time when we take samples actually breaking it into pieces, saving some of it for follow-up immunohistochemistry or for whatever your favorite staining is, taking some of the bulk, just lysing it immediately so that we understand what the population looked like, and then carrying through our cells through processing, making a population there, and then doing our singles. In that way, we can make sure that our singles at the end look like the population that we put into our single cell processing, that we haven't lost a lot relative to pre-dissociation, and that if we find anything that's interesting, we can go back and validate it in the context of the original sample.
Thanks. So my perspective is a little different. My role at Merck is to lead our translational research efforts in immuno-oncology, and so in terms of the technical details, I'm a little less eager to offer a detailed opinion, but I can tell you from a high level, so we're trying to use single-cell techniques to understand the tumor-immune interface in the immuno-oncology setting, and I think that because of the challenges that are inherent, we have to think very carefully about the contexts where doing single-cell analysis is going to add maximal value to our work, because the constraints that are currently in place means that this is expensive, and therefore we can't do it everywhere, and we really do need to work in settings where we can very rigorously control the flow of tissue, and finally we need to do this with the right informatic approaches to really get the kind of data quality that would be strategically actionable out of this type of analysis. And so some of the contexts where we are trying to focus are really around getting information that can then be reduced to practice in the setting of higher throughput techniques. And that includes things like understanding either single-cell signals or cell-cell interaction signals that can then subsequently be deconvolved out of bulk data, RNA-seq or other genomic data, understanding heterogeneity and tumor immune evolution, and how longitudinally immune escape from, or tumor escape from immune pressure might occur. And then I think probably most importantly, understanding drugs in this space where there are pleiotropic effects on multiple cell populations, and where we want to actually understand whether there are potentially both positive and negative effects of a particular drug or drug combination on the tumor cell versus different immune cell subsets within the tumor microenvironment. So that's a bit of a tangent from your original question, but I have to speak to what I know.
Yeah, I think, Peter, there's really two things that are most important. One is your area of expertise, is we need to scale up. It's not just thousands, it's tens of thousands, hundreds of thousands. I mean, we're going to reach billions and billions of reads with Illumina. So scaling up is personally, I think, one very important thing. But I'm not worried about it, because I know if I put you in a room for like a month without writing grants, you'll figure it out. So the field will figure it out sooner or later. What's more of a concern to me is what are the cells that we are going to analyze? What are the cells that we get from patients to analyze and figure out the predisposition to cancer, the therapeutic resistance of these cells? We need to develop minimally invasive ways of retrieving these cells. There's only so much that you could find in the blood. There's definitely a lot more to be explored, but the problem then becomes sort of an ethics problem, is how do we approach it? These are patients. It's no longer research. This is patient material that we're talking about. And for that, perhaps, recently when I joined Memorial Sloan Kettering is where my mind was just absolutely expanded. Clinicians know about these things and they're developing these sort of things that researchers like us do not know about. So the thousands, millions of single cells will happen in the future, no doubt about it. But figuring out exactly how to minimally invasively retrieve the correct cells, I think is something more tricky and perhaps will require a little bit more work.
All right, so my job function right now, I'm pretty new to my function right now in Stanford, and I'm focusing basically more on macroscopic technology. So how we approach like biobanking, how we approach patients' information, how we actually connect all of these dots together, because we will work with massive amounts of data in the future. And applying single-cell technologies will still have a little bit of time because the technologies out there, the variety you have right now is huge, from droplet technologies to microfluidic device, which have all their advantages and disadvantages. And on top of it is when you're looking for the right biopsy, for example, we have to set up a way that we can compare architectures within one patient, for example, to see how tumors basically evolve over time. So we don't have any sample prep or technologies right now who's looking up into the macroscopic technology, like how is the architecture involving in all the genomic data, and how we start actually tracking these data and have an ID on it. It starts with the patient ID, but it goes back and forth into biobanking and comparing data, be collaborative, and so that's my starting point right now, and it's very challenging.
Yeah, well, I've got a whole bank of questions for this panel, but I do want to give the audience a chance. Does anybody out there have anything they'd like to ask? Okay.
Perhaps one thing that Annette brought up is the architecture. I mean, we're all doing single cells.
Yes.
But we also want to learn about the architecture and the proteomics. And the proteomics is where you get involved, Peter. But the architecture is when we get to in situ sequencing, like George Church or Jay Lee are doing. This is also another frontier that we really have to get into, right?
Yeah, so you totally read my mind. Actually, that was really one of the next questions I wanted to get at was, we've heard a lot today about single-cell omics, but most of these are sort of offline technologies, right? Where we have to get, individual cells isolated out of the context of their, for example, native tissue environment, and then we do our measurement. It's usually a sequencing-based readout. But I think there's a sort of competing paradigm where you actually go in and do some type of large-scale imaging approach to getting, in some cases, even sequence information from individual cells in an intact specimen. I wonder if anybody has, I kind of think that may actually be the end game in the long run, but I'm not so confident. I wonder if anybody has any feedback on that.
I feel like you're just trying to be provocative, which is okay. So, personally, I think that the architecture of the tissue is incredibly important. Understanding where cells sit and who they're next to and how that influences their decisions is incredibly important. It's all about intercellular circuits. It's not only about what's inside of the cell. We know that cells behave differently depending upon where they are in your body. People have been doing this for a long time. And so when you dissociate, you lose a lot of that information. The problem with thinking about in situ techniques is it's very often hard to get quite the same resolution on the number of probes that you get. And also, it can be very laborious and slow to image across the tissue and to do the registration. Personally, going back to this idea of what do you do with samples when you're processing them, I kind of feel like there's going to be a hybrid model where you take portions of tissue, dissociate it, sequence it very rapidly over a flow cell, and then use that as a way of rapidly identifying markers that you'll go back and do either targeted sequencing if it's, FoG if you want, or you do your merfish that came out of Xiaowei Shuang's group, or your fish scales out of Long Kai's group. I think that there's probably going to be a melding between the two of these, and I think it comes back to this other question that was brought up earlier. Illumina isn't necessarily going to give us billions and billions of reads all of a sudden that we can use our data for. So an important question becomes, what is the most effective way of using single-cell data? And this is something that David touched on. So I think that using single-cell data to guide selection of probes or to guide subsequent downstream validations that you're going to go after is incredibly important. So in a way, this idea of space, almost maps into some of these other things like perturbations and other variables.
Thanks. So we're very interested in geospatial data linked to high dimensionality data sets around single cell data. But obviously, we think in a very strategic context of where would that actually be important? And does it really require a single integrated technique that can look at all that information at once, or can there be a combination of techniques to answer that question? So in immuno-oncology, we care a lot about the distribution of the immune cells, and particularly whether there are T cells and other immune cells penetrating into the tumor center versus sitting at the periphery of the tumor. We care about the tertiary lymphoid structures around the tumor. And it would be obviously great to do that, to be able to interrogate all of that and how it relates to single cell states. But it also might be possible to do limited sampling, and dissection techniques or other approaches to actually get at those different regions and then just perform non-geospatially resolved single cell analysis on those separate regions, rather than just having a continuous integration of data. So I think we're still very early in thinking about applications from at least an industry perspective around strategically actionable decisions that could come off of that type of a platform. But we're monitoring it pretty closely.
Let me just throw in one more thing for you guys to comment on as you go down there, because I actually am interested in everybody's opinions. One of the problems with single cell analysis is that when you fix the cells, you're getting a snapshot. And so we spend a lot of time thinking about how to build trajectories out of data. But the same thing is true when you think about fixing and looking at a tissue. At any given moment, a cell is in a location. You're all sitting here in this room right now, but it doesn't mean that all of you are from Boston. And if I was to do an analysis over this room, I might find things that are based in Boston, but I might find things that are different. So I wonder too, how much we need to understand the actual, microarchitecture versus sort of the macro architecture of tissues, and what are the right length scales to think about in terms of cellular interactions in neighborhoods?
Okay. I'm not sure I get exactly what you're talking about. But when you're fixing cells, you're fixing a random distribution of cells, random cells. So they're not at one space or one particular trajectory. They're all in different trajectories. And then with the informatics, you could order them, right? So we fix this particular tissue. This cell over here is not the same as cell over there. They're in a different pseudo-time in a fashion because they're all proliferating, right? So you can, using algorithmic approaches, order them. You don't need to synchronously define a particular population and then let it go. Assume, for example, you're talking about TGF-beta treatment for a particular synchronous culture. If you have a particular tissue where you know that there's signaling, TGF-beta signaling or whatever it is, the cells that you get from a tissue are a random sample, and all these cells are in different time continuum, right? From 0 to whatever time it is. That's my personal opinion.
Yeah, that comes to my comment how we standardize all of these, because if we at one time try to, go into diagnostic or make actually something useful for the patient, so we can go more into personal medicine, which is basically what it is. If single cell is the answer for cancer, then we have to be able to standardize all these methods. And right now we have how many methods actually out to just do mRNA, or what kind of different sample prep we have. So it becomes very, very difficult to standardize and basically to compare the data, even, there might be the same cancer, but even, having the same cancer could be, we know the heterogeneity is very high. So standardizing all the methods, it's going to be the challenge for the next couple of years. And before we're not getting this together, it's not going into the clinic.
So I guess to that point, how important do you guys actually think it is to standardize the methods? I understand that if you have one population that's done in one method and another population that's done in another method, they're not comparable. But if you have internal comparisons, say multiple different states that exist in the same cancer, different clones, you can do a differential expression analysis, which is almost like thinking about things in terms of qPCR, back when we used to do delta-delta CT. And the question becomes, will you find the same patterns? And I think in a lot of the data that we've done, we have. So, if you're finding these pathways that are enriched and you assume that you lose some transcripts, but you're still getting a lot of the same information. So do you think that it's essential that we standardize or that we better understand how to structure the analyses in the front so that the comparisons are more fruitful?
Yeah, that's one part, but on the other side, also, we need to have a way to communicate these different platforms, right? So there's the architectural side where we need to know the exact location, the time, how cells evolve in a tumor, and how we can, approach this problem. But the other side is the sample prep, which is, so it becomes convoluted if we don't find standardization measures, which could be something, an internal control. So yeah, that's.
I mean, I'll just give my 2 cents. I'm technically maybe not supposed to say so much, but I think what we really need to standardize is basically performance, right? So once we've sort of standardized performance, it doesn't matter whether you're using, SmartSeq or CellSeq or whatever, right? If you can sort of get to the same coverage per cell, I think I kind of agree with Alex at the end of the day. Two people on two different sides of the country better see the same sort of subpopulations when they dissect the same tissue specimen, right? So at the end of the day, it's the interesting thing about single cell is it's not so much the expression level of each gene in each cell that's the bottom line, right? It's what's in this mass of tissue, what cells are actually there, and it's in a sense easier to boil down. When you think of it in terms of cell states or cell types.
Yeah, and again, speaking from my bird's eye view here, what seems most important is really the bioinformatics, and what you're talking about is essentially reducing the dimensionality of the data through the right bioinformatic approach so that what you're looking at is not the noise but the very robust signals and the meaningful signals. And obviously the other aspect of the bioinformatics is the QC that's inherent in that.
I agree with the bioinformatics part, certainly, that we're going to have to refine our bioinformatics methods. But also bioinformatics can only work to a certain level, right? Like microarray technologies could never tell you the stuff that next generation sequencing technologies are telling you. Not always, but in certain cases. So we mustn't forget that we need to better the current technologies, and I'm sure we will. And the marriage between the bioinformatic analysis that will keep getting better and better and better, and the methods to actually develop to obtain the data are going to evolve more and more.
So I guess the big question to me becomes, is the next step in improving the analyses to just collect better RNA sequencing data that's more consistent, or is it really about trying to couple together sort of a more multi-omics approach where you can, is it more important to measure the epigenome, the proteome? Is it more important to be able to couple those things back together and be able to use data that people already have as a way of understanding the patterns that exist in your data?
You're absolutely right. The more modalities we test, the more associations we can make, the more confidence we get in our data, right? That's
but
essentially
what's most important? What is the next step? Because I'm not sure it's another 10,000 cells. I think it might be measuring protein and RNA at the same point, or being able to link DNA mutations directly to RNA profiles.
Okay. Well, assume cancer as an example. We know that mutations resistance can come from a cell that's found like 1 in a million or something like that. So that argues that perhaps doing 100,000 cells is useful if you can observe that one single cell, right? It's always context that matters. If you're doing immunology where you're looking at subpopulations of T cells, which ones attack the tumor better, things of that order, perhaps you don't need that very fine grain analysis where you delve into hundreds of thousands or millions. It always depends, we're scientists. It always depends on what sort of question, clinical or biological, that you're asking.
I would argue for just not having that discussion in a vacuum, but rather thinking about it in terms of specific use cases and how you would actually use the data. I think the idea that truth will just emerge from enough data is one that probably none of us really believe, and we all I'm sure think that we need to bring some very specific directed questions to this type of data. That being said, you could probably make an argument for a number of different ways to go based on a lot of really interesting data that could come out of each of those tool sets. So it's hard to answer without having a much longer conversation.
What would you guys like to see? We're biologists and we develop technologies. We're clinicians. We are looking for minimal residual disease, whatever it is. What do you guys want us to develop, right?
A brave soul.
What I would really like to see is that I can extract RNA from a cell and just directly throw it onto a sequencer. It could be a nanopore or something, which tells me the sequence of the RNA, the full length, a long sequencer, and also the modifications that are there on the RNA molecule. If I'm coming from an RNA background, so I'm talking about that. Can we get that, at least for a start?
So what's the dissociation constant of the nanopore, right? If the I've actually thought of the same thing. Can we squirt a single cell lysate into an Oxford nanopore machine and get a transcriptome or maybe a genome? I haven't tried, but I'm guessing that's kind of what you're thinking, right? Something along those lines, right?
Can I comment on that? So is it more you worried about that you directly can sequence without any amplification and putting more bias into, yes, you're going this way?
So I would like to cut out the reverse transcriptase, cut out the polymerase for PCR.
Yeah, I think that's a wonderful idea. I think we're actually aiming to this goal. Except nanopore, I don't know what technology can do this right now, but then we have only one cell, so we still need to multiplex and to look into like thousands of cells really to understand the biology, right? But I know where you're going to, because we're probably missing a lot, and that's why I actually pitch into the standardization. So there are lots of methods we add more bias to, like especially in RNA, we amplificate a lot of RNAs we already know, and so that actually also applies for single cells or, compared to bulk sequencing. Yes, I think that's actually the approach to go, more likely, but there are no technologies right now out there.
I guess my question would become, if you're going to try and go directly, why would you not try and profile directly in situ? So I think going back to the technologies that we already talked about, what is the point of dissociating and breaking apart? If the ultimate goal is to understand all these things and understand them in situ, really do we just need to figure out better probes for going in and looking at different modifications as they lay? Because even if we were to pass through a nanopore, we're going to have to figure out how we measure each of those modifications. So there's going to be some sort of chemistry. Why divorce it from the context of the cell? Why throw away information when you potentially could? So as long as we're going to dream big, might as well do it all exactly where it lays and get all that additional information. But I think what you're bringing up is something that we were talking about before. It's all thinly veiled. It's what is the most important information to collect? What is the order you should collect it in? And what is it that you need to address your specific problem? Going back to David's point that you can't say that one approach and one set of variables being collected is superior for everything. And so in your case, the question might become, what is the trade-off between getting base specificity versus being able to look at multiple different transcripts together? So I don't know the answer, but I'm sort of curious to know what everybody thinks is the next thing that we should be developing. But maybe it's RNA modification readouts.
Any other brave audience members? Okay.
You guys are already articulating a question that I had coming in here today, but so my question is, if you had to choose one thing today, based on the current limitations of NGS sequencing reads, the number of reads from a single run, would you choose targeting, let's say, 10,000 targets from a limited number of cells, or targeting a limited number of targets from a lot more cells? So is the number of targets more important, or is the number of cells more important today, given the current limitations?
My answer is that it depends on how heterogeneous that cellular population is that you're looking at. So, if it's a very heterogeneous population, maybe a complex tumor specimen with lots of interesting immune infiltration and other things that are important, I'm going to go with lots of cells over lots of targets probably as my first swipe to get a lay of the land. And it's from there that I'm going to try to figure out maybe what targeted approaches I could follow up with, I guess.
I think I might flip the question differently, because what you're really asking me is how much information do I want to collect from any given cell? And so in the selection of transcripts, I'm assuming that I have knowledge about what's important in the system. So the question really to me is, do I sequence it shallowly and get an idea of roughly what it's doing or what it's expressing highly, who it thinks it is? Maybe thinking about some ways of depleting out things that are very highly expressed that carry limited information content and use it that way? Or do I sequence things more deeply? And I think that it's becoming increasingly clear that having a shallower understanding of more cells, but understanding how they clump together and getting better resolution is great, because it removes some of the technical noise associated with every single prep and some of the stochasticity associated with the biology. And so I would almost argue that doing shallower sequencing rather than trying to target it is a better way of using bandwidth, particularly as you increase scale. And that's not perfect everywhere. And I think in places where you really want to get some information, say, for example, you're a TCR person and you want to actually just read out the CDR3, I think more directed things that give you the information that you want is probably the best way of going after it.
Yeah, again, I think it depends on your use case. So, if you're looking for essentially dynamics of different cell types and perhaps some limited phenotypic information, which may in many cases be really what you want to do, then I think going less deep is appropriate. But if what you're really trying to do is to, in an unbiased way, reconstruct the intricacies of cell-cell interactions to try to make some inferences about the biology that's driving a certain phenotype, and if you were trying to find nodes that are most targetable based on the non-redundancy, then that type of exercise you would really want to go deeper.